Hi everyone. Today we are going to talk about osmotic pressure and its role in a plant life, using tomato as a representative example. The goal of this presentation is to explain osmotic pressure. Yanlış söyledim ya. Çok özür dilerim. Tamam bir daha.
Kanka bunlara
Bir daha.
takılırsan devam edemeyeceğiz ama.
Tamam çok haklısın da çok yanlış söyledim çünkü. Okay. Hi everyone. Today we are going to talk about osmotic pressure and
İsim,
its role in plant.
isim, tanıt.
Okay. Hi everyone. Today Ahmed and me are going to talk about osmotic pressure and its role in a plant life using tomato as a representative example. The goal of this presentation is to explain osmosis from a thermodynamic perspective and then show how this concept becomes essential for the structure and survival of plants. Let's move on. Contents. These are our contents. We are gonna explain firstly what is osmosis, then thermodynamic driving force, then equilibrium and osmotic pressure, why osmosis is essential for plants, tomato fruit, chemical composition of tomato fruit, estimation of osmotic pressure for tomato fruit cells, physical meaning of osmotic pressure, and final part is conclusion. What is the osmosis? Osmosis is defined as the net movement of a solvent, typically water, across a semi-permeable membrane. This semi-permeable membrane is a selective barrier that permits the passage of the solvent while effectively restricting the passage of dissolved solutes. What about driving force? Well, the driving force for osmosis is the difference between chemical potentials through this membrane. The chemical potential of a substance in a solution is a measure of its free energy per mole, and the system naturally evolves in a direction that minimizes its Gibbs free energy. When there is a concentration difference across this semi-permeable membrane, the chemical potential of water is different on each side, and water moves in order to reduce this difference. Let's move on. Fourth slide, please. The presence of solutes in a solution reduces the chemical potential of the water compared to pure water at the same temperature and pressure. This reduction is primarily an entropic effect. The mixing of solute and solvent increases the overall entropy of the solution, which lowers the Gibbs free energy and the chemical potential of the solvent. Let's go a little deeper into the mathematical structure of this concept. Could you make me host, please?
Yes, you can continue.
Okay. Give me a second. I'm gonna share my screen right now. Can you see my screen?
Yes, we can see.
Okay, very well. Um, Everyone knows fugacity concept. We use these concepts through the semester, using the fugacity to represent the chemical potential of a liquid solvent. Then we have chemical potential of water is equal to chemical potential of water in a standard state plus RT ln fugacity of water. Let's highlight it in a color. The equilibrium condition reduces the equality of solvent fugacities. Then we have fugacity of water in first cell is equal to fugacity of actually partial fugacity of water in second cell. For the solution side, the fugacity of the solvent is written as fugacity of water in second cell is equal to the mole um, the mole of um, the mole fraction of solvent in second cell multiply activity coefficient in second cell multiply fugacity of water at this temperature and pressure. Let's highlight it in a pink color. Okay. This expression clearly that the presence of solute reduces the effective thermodynamic escaping tendency. Escaping tendency of the solvent through a decrease in solvent activity. Because the membrane is rigid, the pressures in the two components needed to be equal. Therefore, the pressure dependence of solvent fugacity must be included. For a liquid solvent, the fugacity at two different pressures is related using the Poynting correction. Everyone who attended these courses regularly remember this term because it was many times explained by Deniz Hoca during this semester. Using this Poynting correction, we have obtained fugacity of water temperature at this temperature and pressure in second cell is equal to fugacity of water at this temperature and pressure in one first cell multiply exponential molar volume of water or molar volume of solvent, but typically, we use water, liquid in a liquid state, and pressure difference over, of course, RT. Um, this one is represents. I'm gonna use different color. This term, molar volume of water. And we have some assumptions about this molar volume of water, assuming constant volume. Constant volume. Why? Because of incompressibility. Then we have pi. That means pressure in second cell minus pressure in first cell is equal to minus RT over molar volume of water in the liquid state multiply ln molar fraction of water in second cell multiply activity coefficient of water in second cell. This result demonstrates that osmotic pressure arises from the reduction in solvent activity caused by dissolved solutes. In the limit of dilute and nearly ideal solutions, where activity coefficient of water in second cell approximately 1. Let's write it on our pages. In the limit of, let's highlight it as different color. In the limit of dilute and nearly ideal solutions, then we can assume activity coefficient of water in second cell approximately equal to, 1. And the mole fraction can be approximated. This equation reduces to 1. Let me use different color. 1/2 relation. Most of you are familiar with this relation, I think. And now I would like to briefly explain why the natural logarithm and its Taylor expansion are used in the derivation of osmotic pressure. In general, a Taylor series is a way of approximating a function around a reference point. For a function f of x, the Taylor expansion around a point, let's say x0, is written as f of x is equal to f of x0 plus first derivative of f of x0 multiplied x minus x0 plus second derivative of f of x0 over 2 factorial multiplied x minus x0 square plus dot dot dot, it goes to infinitely. If this equation is taken around x0 is equal to 0, it's called a Maclaurin series. You can remember this series from 119 or 120 course in the Department of Mathematics. Now, consider the natural logarithm function and to approximate the logarithmic term appearing in thermodynamics, we expand the function of ln 1 plus x around x is equal to 0. The Taylor or Maclaurin expansion of this function is ln 1 plus x is equal to x minus x squared over 2 plus x cubed over 3 minus x to the power of 4 over 4 plus dot dot dot. This expansion is valid when the magnitude of x is much smaller than 1. We can say this expansion is valid. In osmotic systems, we encounter the term ln, let me write it, ln 1 minus absolute. Absolute represents the solute of mole fraction. And since the solution is dilute, absolute is a very small number. So substituting x is equal to minus absolute into the Taylor expansion, then we have obtained ln 1 minus absolute is equal to minus absolute squared over 2 minus absolute cubed over 3 minus dot dot dot goes to infinity. At this point, the series can be reduced. Because absolute is much smaller than 1, high order terms such as x squared over 2, um, x cubed over 3, x to the power of 4 over 4. These terms are actually, we can say, this term very small number. So we can neglect these terms. Why? Because if you compare the first order, that that is the x first order, this one, this first order term, this one, and these are the rest of the expansion. But this rest of the expansion is very small number, so we can neglect these terms, and we have approximately ln 1 plus x is approximately equal to x. So we have 1 minus absolute is approximately equal to 1, minus absolute. This is known as a first order Taylor approximation. At this stage, osmotic pressure is written as pi, which means the pressure difference between two cells minus RT over molar volume of liquid solvent, let's say solvent, multiplied ln x solvent. So this is our equation. Okay. For dilute solutions, the solvent mole fraction is close to unity. Therefore, we can apply a first order Taylor approximation to the logarithmic term. Using this approximation, we have obtained x solvent plus x solute is equal to 1. I'm gonna rearrange this equation again. Then x solvent is equal to 1 minus x solute. So we have ln x solvent is equal to ln 1 minus x solute is equal to, by using this approximation, we have obtained minus x solute. And then let's rewrite our pi. Our pi is equal to minus RT, molar volume of solvent, of course, in a liquid state, multiplied ln x solvent. Then this term approximately minus RT molar volume of solvent again, then ln x solvent is equal to 1 minus x solute from this equation. By the way, I'm gonna add some a little bit information on our right side of the page. We know that x solute, maybe zoom in, x solute is equal to moles solute over mole solute plus moles solvent. We know that this term is very small number. Ahmed is gonna explain our representative example of tomato in on upcoming slides, and we are gonna see our percentages of solutes in our tomato. Probably it's gonna be percentage 5, less than 5, or about 5%. So compared to mole solvent, mole solute is really small number, very small number. So we can, of course, we can neglect this term. So therefore, X solute is equal to moles solute over moles solvent. What does the mean? Sorry. Moles solute is concentration of solute over molecular weight of solute. Moles of solvent is equal to concentration of solvent over M solvent. I think here is clear. Then let's go back to our main equation, pi. So this pi is equal to minus RT, molar volume of solvent. This term is equal to actually minus solute by using a first order Taylor expansion. We have obtained minus X solute, and then I'm writing this term here. So again, this is approximately equal to RT X solute over molar volume of solvent. Then we have already found X solvent is equal to this one. I'm gonna substitute this term into the here. Then we obtain RT and okay, C solute over M solute over volume of solvent multiply C solvent over M solvent. We have already known that this term is equal to what? 1 over, let me write it in different color, this one. So, and then these two terms actually cancel out each other, and then it is the pi is approximately equal to RT C solute over M solute. So finally, pi approximately RT C solute over M solute, molecular weight of solute, of course. This is final part of my approximation by using Taylor, first order Taylor expansion and the logarithmic term. So I can stop here. Ahmed is going to explain the rest of the presentation. Ahmet, you can continue with another
Thank you
slide.
for your explanation, Zeynep. Now I will continue. I will share my slides right now. Now I'm using tomato Solanum lycopersicum as a simple way to explain osmotic pressure in plant life. Tomato is a great example because we all know how it behaves. Sometimes it feels like a ferment fresh, sometimes it becomes soft, and when it loses water, it can shrink and wrinkle. A big part of that is the most important part of this is water balance. Also, tomato fruit is basically a water-rich, around 95% water by weight. So even though it looks like just a soft fruit, it's actually a system where water is the main component, and dissolved substances inside the cells become extremely important. Those dissolved substances are what create the osmotic effect. Now we will look at the chemical composition of the tomato fruit. I will show the approximate chemical composition of a tomato fruit. The main message is very clear. Most of it's water. 95% of the tomato plant is water, and the remaining part is the dry mist. Inside that dry mist, the most important group for osmosis is sugars, especially simple sugars. Then there are smaller contributions from proteins, fats, and other compounds like acids and minerals. These numbers can change from one variety to another and with ripeness, but for our purpose, we don't need perfect precision. What we need is the idea that tomatoes has contain a meaningful amounts of dissolved solutes, and those solutes strongly influence how water moves in our fruits. Estimation of a osmotic pressure for tomato fruits. Now, this part is a rough estimate, more like an engineering approximation than an exact biological analysis. A tomato contains countless different molecules, so we can calculate osmotic pressure perfectly would be very complicated. So we can simplify. We can assume that the main osmotically active part of the dry mist is sugars, and we treat those sugars as if they behave like a typical simple sugar. This is not perfect, but it gives a rough scale. With that kind of an approximation, the osmotic pressure of tomato fruits comes out around 0.8 megapascal, and that matches a typical range reported for fruits, something like 0.2 0.9 0.3 to 0.9 megapascal, depending on variety and ripeness. Decay point is not the exact number. Decay point is the scale, because soft fruits like tomato can have an osmotic effect that is surprisingly large. Physical meaning of this osmotic pressure. What is this? Now, the most important clarification is this. Osmotic pressure does not mean that the tomato is living in a high pressure environment. The surroundings are still around 1 atmosphere. Osmotic pressure is basically a way of expressing how strongly dissolved substances inside the cells pull water in. Water tends to enter because the inside contains more dissolved solutes, but plant cells don't keep swelling forever because they have a cell wall. The cell wall resists expansion. So when water enters, The cell builds up internal pressure. That internal pressure is what we call turgor. This is why osmotic pressure matters biologically. It helps plant tissues stay firm and supported. Second, it contributes to growth because cell expansion depends on having enough internal pressure. And lastly, it helps the plant keep water and keep functioning when conditions become drier. To conclude, osmosis in plants is really about water moving in response to differences in water's chemical potential, not just as a simple concentration rule. Inside the plant cells, the salt wall solutes creates a tendency for water to enter, but the cell walls prevents uncontrolled swelling. This turgor is essential for plant structure, growth, and the regulation of water movement in tissues. In tomato fruit, the balance between solutes and water strongly shapes firmness, texture, and water retention. So the main takeaway is that osmotic pressure is meaningful even under normal atmospheric surroundings because it reflects the plant's ability to control water movement and maintain internal support. These are our references. Thanks for listening.